Thank you for the introduction. My name is Keenan. I'm in the Division of Bioequivalence 3, Office of Bioequivalence within OGD. Today I'm speaking about identification of research opportunities for ANDA submission related to bioequivalence for orally inhaled drug products. Here is my outline. First, overview of FDA recommendation on bioequivalence assessments for orally inhaled drug products, OIDPs. Next, alternative B approach to comparative clinical endpoint B study for solution-based middle-dose inhaler, MDI. During our ANDA evaluation, we received some applications use alternative B approach for suspension-based MDI. We will discuss additional considerations and further research for using this approach for suspension-based MDIs. I will also share additional consideration for formulation sameness. Lastly, I will provide a summary. Developing generic locally acting OIDPs is challenging because of the multiple factors that can influence drug delivery to the site of action. For example, the interaction between formulation and the delivered device can influence how the aerosol performs, which can impact regional deposition. Subsequently, the solution of deposited drug, the patient and the device interaction, which can consider complexity of using the drug product, can impact those of administration, which also influence the regional deposition. All the factors together can influence overall absorption of the drug once deposited at the site of action. To address challenges for locally acting OIDPs, the current method being used is weight of evidence approach. It includes in vitro BE study, in vivo PKB study, comparative clinical endpoint BE studies, formulation sameness, and device similarity compared with reference product. With this approach, each of 4 components need to meet bioequivalence criteria. There are so many challenges surrounding clinical endpoint study in the weight of evidence approach to make it difficult to establish bioequivalence for middle-dose inhaler and dry powder inhaler, such as higher variability, lower sensitivity for evaluation of formulation difference, longer study duration, and more costly than other types of bioequivalence study. Therefore, alternative bioequivalence approach can be used to address those challenges. As you can see, the local delivery of API to the site of action is a complex, multiple-step process with each step impacting the next. The alternative approach for OIDPs should consider all those steps. Alternative approach to the comparative clinical endpoint study has been published in several PSG for solution-based MDI, including dexamethasone middle-dose inhaler. As recommended in the PSG, if our generic show formulation sameness and the device similarity to the reference MDI, additional supportive study may provide a foundation to help ensure the equivalence at the site of action. Those additional studies include the following: characterization of emitted spray, velocity profile, and evaporation rates, which could help achieve to understand drop size and the evaporation process of the formulation emitted from device. Morphology image comparison, characterization of a full range of residual drug particle size. Another supportive study is to use more predictive APSD testing by using representative mouth sort-through model and breathing profile. This can help to capture and understand impact of patient variability on the drug deposition. In vitro dissolution study can help to understand how API drug particle dissolved at the site of action for absorption once deposited within the lung. Quantitative method and modeling, such as physiological-based PK, computational fluid dynamic study, can be utilized to provide the data on IV-RVC to help bridge gap between in vitro product performance and original drug deposition. Alternative PK/BE study can be considered to understand how PK study may correlate to local deposition. So, when considering alternative bioequivalence approach for suspension-based MDI, the difference in drug delivery process compared with the solution-based MDI, particularly the effects of suspended API particles in the formulation should be considered. For example, may include understanding surface level interaction that could impact downstream process critical for regional drug delivery, such as van der Waals and electrostatic force can lead to change in API PSD over time through agglomeration. Surface roughness of the API particle may impact stability of the suspension. Interaction of API with the exhibit used for formulation stability, which may lead to difference in PSD of dry particles. To address those factors, characterization of suspension-based MDI pre-actuation need to be considered. So as our hypothetical example, in addition to the study discussed earlier for the solution-based MDI products, characterization of the effect of suspended API particle and interaction with formulation on the drug delivery process could also be included in the alternative bioequivalence approach for suspension-based MDIs. Please keep in mind, it's important that alternative BE approach include a study adequately address how those suspended particles impact each aspect of drug delivery process. It's highly encouraged that applicants submit their proposal for alternative BE approach on suspension-based MDI through pre-NADM meeting request. Here is some general consideration for alternative BE approach for OIDPs. Approach should address sameness of delivery at the site of action. Approach should be scientifically justified with our comprehensive data and explanation. Due to the complexity of many different factors that can affect generic drug product performance, critical key attributes for suspension-based MDI may be product-specific. It's important to understand key quality attributes of your generic product in compared to the reference product that will influence the in vivo BE study. Next, we will discuss formulation sameness. Even though there's no requirement for Q1, Q2 the sameness for certain product, specific bioequivalence approach may be recommended in product-specific guidance for generic Q1, Q2 formulation, such as oral suspension locally acting drugs, and OIDPs. A longitudinal application with no regulatory requirement may be submitted to FDA for OIDPs. However, sufficient data and information are needed to justify the impact of formulation difference on bioequivalence and safety. The first longitudinal MDI product was approved this year in March. If the applicants want to develop their product, not Q2 as the reference product, bridging study should be submitted to demonstrate that formulation difference does not affect product performance in the submission. For example, evaluate the impact of different amount of the proposed exhibit, testing of multiple drug-to-exhibit ratio that encompass combination below and above the ratio in the proposed formulation on drug performance. Please keep in mind that some change in exhibit level may or may not impact product performance. This could be considered as a case-by-case basis. In summary, OIDPs are complex drug-device combination products with multiple factors contributing to their performance. The type of study include as part of Alternative-B approach to the clinical endpoint study will be product-specific, as difference in dosage form and formulation will give rise to a different area of uncertainty. Applicants are highly encouraged to submit a pre-NDA product development meeting request and seeking the agency's feedback on your proposal. Approach should be scientifically justified with our comprehensive, significant body of data. I would like to thank Passer, April, Bing, Utpal, Brian, Susan, and Liangfang to help me develop this presentation. Thank you for your attention.